Hello, and welcome to our second lecture in our Microbial Genetics lecture series. Today we'll talk about the structure and function of DNA and really see how DNA is the fundamental unit of this chapter. Because without DNA, we won't be able to store the information, and without storing the information, we can't pass on our information to complete our central dogma of DNA to RNA to protein. So while protein is the workhorse and RNA is literally the messenger, DNA is the storage bank of all of our genetic information. So how does it store that information? Well, it stores it in the fundamental unit of DNA, which is the nucleotide. Okay? The nucleotide you can see in figure A there. It is consisted of a phosphate, a deoxyribose sugar, and a base, or nitrogenous base. Okay, you can see nitrogenous base right there. Okay, this nitrogenous base is adenine, or A. Okay, and we'll see in the next slide that we have 4 different nitrogenous bases that we can choose from, and how they are arranged is what gives us our genetic code. But before we get there, we want to talk about deoxyribose. Right? We learned about sugars in our microbial biochemistry lecture series. Right? We were learning mostly about glucose and how that is a hexose, right? Or a 6-carbon sugar. Here we see we have a 5-carbon sugar. Okay, 1, 2, 3, 4, and 5. Okay, as you can see, you can count them as well, and it counts it for you. 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. Those are called primes because the other carbons in the base will be numbered as well. And so the deoxyribose sugar is denoted with that tally mark prime, so that you can count those separately from the base. Okay? And remember, we talked about phosphate groups and how ATP and the like is necessary for metabolism, and so that same phosphate group is on the backbone of this whole thing. It's even called the phosphate sugar backbone. Okay, and so that phosphate group is actually there from ATP and the like in its counterparts as it actually gets attached to the strand. So, without further ado, the 4 bases. Okay, we already saw adenine. Okay, we have adenine and guanine. These make up our purines. Okay, and then our cytosine and thymine. Those make up our pyrimidines. And interestingly enough, a pyrimidine and a purine pair with one another on complementary strands. Okay, so an A and a T will always pair together. A G and a C will always pair together. All right. Now, pyrimidine and purine are 2 terms that you'll want to know in the sense that A and G are purines, adenine and guanine, and C and T are pyrimidines and cytosine and thymine. Okay, but how do you memorize which is which? Okay, I like to memorize it as adenine and guanine have 2 structures, right? Okay, so they have 2 structures compared to 1 structure for cytosine and thymine. Okay, so I then look at the name pyrimidine and I think, Okay, well, pyramids are complicated and complex, and it doesn't make sense that these complex guys are called purines when the pyrimidines look more pure and the pyrimidines look more complex. Okay, so I know it's kind of like a flip-flop way of thinking about it, but it's the easiest thing I can come up with in my head. Feel free to share a example that you might like. But remember, C and G and A and T, those always go together. Okay, so purine and pyrimidine are always together. Okay, and how I said, this sugar phosphate backbone, what the heck? When you want to join a couple of bases together, nucleotides together, you are going to need energy from somewhere. You're going to get it from that phosphate group on the end there, okay? And you're going to attach along the base. You're going to see that there's a sugar and a phosphate on the outside, leaving the nitrogenous bases to pair up on the inside, okay? So this is the backbone, sugar phosphate backbone, SP backbone, and these are the bases inside. Okay, this is backbone. Okay, so we also talked about Chargaff's rules in a bit. The scientist Chargaff found that when he purified DNA, no matter what, the amount of A and T and G and C always differed from species to species, but the amount of A and T was always equal. The amount of T and G was always equal too. Okay, so if you had 30% adenine, you had 30% thymine. Okay, which leaves 40%, which means you have 20% guanine and 20% cytosine. Okay? It's just the math. It's always going to work out that way. So if you know the amount of adenine, you pretty much know the amount of all of the base pairs. Okay? And we can't mention the backbone and the structure of DNA, obviously, without mentioning the two, as it says, pioneering scientists, okay? James Watson and Francis Crick. Okay? They all led to these two men put together the structure of DNA for the first time. But we can't go and say that without mentioning that they had the help of Rosalind Franklin's X-ray diffraction pattern of DNA. Okay, and Watson and Crick were lucky enough to see it before she published it, and then they were able to use it to, you get it now. And so without this famous picture here, they wouldn't have discovered the way the structure looked. Now they might have done this picture later anyway, but definitely this was their aha moment, and we must credit Rosalind Franklin for helping in the discovery of the structure of DNA. Okay, and remember, this is a time before they even knew what DNA, really was. At a certain point, they didn't know what it was, and they had recently just found out that it was the genetic code in all of us, and it was them that really kind of kicked that off. This kind of pairs it all together. They proposed the double helix model, that it went to a double helix, okay? And that is just based on the shape of how you would organize these bases, right? They're just going to naturally spiral, okay, as they kind of bond and bend and kind of keep the bases toward the middle, right? They're just going to naturally spiral. And so that you can actually measure the amount of major groove and minor groove. And then note A and G bonding together, G and C bonding together, A and T bonding together, G and C bonding together. Okay. And note in here are dots. Okay. You can see the nitrogenous bases A and T. When they kind of pair up, they kind of make two hydrogen bonds across them. Okay. And this is how we describe that hydrogen bond visually. Okay. They're kind of paired up and they kind of want to match up. They don't want to pair up with anything else. They won't pair up with that C, for example. They'll kind of push them away. Okay. And so that C and G pair up with 3 hydrogen bonds. Okay. And that's why they pair up so easily. Imagine coming in with 2. It's kind of not be, as active, as energetically favorable to do that than it would be to just bring the G into alignment with it. Okay? Just how it works. Okay, and so those are called hydrogen bonds, and again, this is how we visually look at it. Okay? Again, A and T and C and G pair up, okay, and they do so through hydrogen bonding. Okay, all right. So that is our structure and function of DNA. We'll come back to DNA after we take a look at the structure and function of RNA. We'll look at how it replicates and then how it makes RNA after that. So we're going to learn DNA, learn RNA, and we learn how DNA makes itself, and then we'll learn how RNA makes itself, and put it together in protein. Okay, see you in the next video.